Welcome again, sqadians. Our topic of discussion today is antibody diversification and synthesis. We know very well that antibodies are actually proteins that belongs to the class of globulins and are mainly responsible for providing immune defense against harmful pathogens and toxins. That's why they are also called as immunoglobulins. These antibodies are the solubilized components and thus aids in humoral immunity. That is an important component of specific or adaptive immune system. When we say antibodies are part of specific immunity, it is because of the fact that it can target specific pathogenic antigens. It recognizes and binds that foreign antigen, and then either neutralize it or generate other immune responses against that antigen, so that it can be removed from the body and no longer can harm the body. Now, what is antibody diversity? That is our main topic today. Well, we know that there are millions of antigens present in our surrounding. And surprisingly, the human body appears to be capable of generating an almost infinite number of antibody, or immunoglobulin, molecules, perhaps at least one for every antigen in the universe. Some calculations suggest that there could be as many as 10 to the power 11 different antibody molecules available in the human body. This vast diversification in the antibody production is called antibody diversity. But we have a general concept that says a single gene codes for a single protein. So if this concept applies here, then there must be billions of genes coding for immunoglobulin molecules. But we know that this is not the case, as the total of around 40,000 genes are found in our genome. That is responsible for producing a variety of proteins, and so among them, only few genes are responsible for coding of immunoglobulins. Then how this diversity in antibody structure exists? Well, we will get to know the answer to this and so many other questions in this video. Also, this process of diversification takes place during B cell development. So we will first make our concept about B lymphocytes development. Then we will be able to understand how B cell achieve this diversity. Later in this video, we will try to understand the basic revision of antibody structure. Then we will learn about gene organization and synthesis of heavy chain, where we will learn the process of somatic hypermutation, VDG recombination, and the process of class switching, and how these processes aids in antibody diversification. And lastly, we will learn about gene organization and synthesis of light chain, including both kappa and lambda. Don't forget to download our apps so you can watch our video lectures anytime, anywhere.